Hi everyone, welcome to another of our urinary system lectures. Today we're going to be following where we ended in our previous lecture. Previously we looked at filtration, where we were removing substances from the blood inside Bowman's capsule at the glomerulus. We were producing filtrate. Now we are going to look at what happens to that filtrate as it moves through the nephron. So, as always, let's start with our not really attendance questions. First, what does ADH do at the kidneys? Next, similarly, what does aldosterone do at the kidneys? And third, what is reabsorption? Go ahead and pause your video, try to answer those questions. Hopefully you've done that, so let's see what the answers are. At the kidneys, remember ADH also causes vasoconstriction, but we're not concerned with that right now. ADH at the kidneys causes them to reabsorb more water. We will actually look at how do they do that a little later on in our next lecture. But right now we will just say they cause them to reabsorb more water. What does aldosterone do at the kidneys? Aldosterone causes the kidneys to reabsorb more sodium. They also cause them to secrete more potassium, but reabsorb more sodium is really the big thing that we're concerned with there. So now we've said reabsorb twice. Reabsorb more water for ADH, reabsorb more sodium for aldosterone, but what does reabsorb mean, or reabsorption? What does that mean? Well, when we're talking about the kidneys, reabsorption is taking something that was previously filtered at Bowman's capsule. It was put into the filtrate from the blood. Well, now we're going to take it from that filtrate and put it back into the blood because our body wants to hang on to it and not remove it from our body in the urine. So today we're going to look at reabsorption and we're going to look at secretion, two things that kind of are the opposite of each other. So as we've done a couple of times now, we're going to take a look at the nephron. Here is kind of an in-between view of a nephron. This is similar to the nephron that I say is the way that we draw it. It's a little bit more complicated, but it's still a very basic nephron. Here's our proximal convoluted tubule, the PCT, and here is the loop of Henle, the descending loop and the ascending loop. And here's the distal convoluted tubule, the DCT, and here's the collecting duct at the end. Now, this is still a very basic looking nephron. It's just drawn a little bit more realistically with the twisty PCT and the DCT. But again, as I always like to remind you, here is that more realistic view of the nephron. Very, very twisted up and surrounded by blood vessels. So as we go through the nephron, which is the yellow part, the tubules, the yellow part, it's important to remember that all of those tubules are surrounded by capillaries, which are the purple parts. Because we are constantly, as we move through the nephron, we are constantly taking things out of the filtrate that is inside the nephron and moving it into the capillary, the purple part. And we are taking stuff from the blood inside the capillary and putting it into the filtrate, which is inside the nephron. So we're constantly moving stuff back and forth between the yellow tube and the purple tube. So keep in mind when we see something that looks like this or that other way that we sometimes draw the nephron, there is a blood vessel surrounding it, like this. That's how that reabsorption and secretion that we're going to talk about today works. We have to have that blood vessel intimately surrounding the nephron to make it really, really easy for stuff to move back and forth. Okay, so first, let's talk about reabsorption or tubular reabsorption. Now, first, let's learn some terms. We're going to talk about basolateral and apical surfaces. Now, what I've done right here is I've just drawn part of a tubule. It does not matter if I'm talking about the PCT or the DCT or the loop of Henle. It doesn't matter. This is just a generic tubule. And right here, this is the inside of the tubule where the filtrate is flowing. And I've drawn the arrow to show you the direction of the filtrate flow, which even that doesn't matter. It could just as easily be flowing in the other direction. Here is one wall, here is the other wall. So I've cut it open so that we can see inside of it. Now, the tubules are one single cell layer thick. So here, this is one layer of cells, and down here, this is one layer of cells, and here, this is hollow. So inside, what would we call this hollow area where the filtrate is flowing? Think of this as kind of a thinking cap question. What do you call the hollow area of an organ where blood or filtrate or whatever would be found? That's called the lumen. So here, this is the lumen of the tubule. Now, up here, this wall, the inside, right here where my laser pointer is, this side faces the lumen, and this other side faces the blood vessels. It faces the interstitial area and the capillary. So, the side that faces the lumen, is the apical side. And the side that faces the interstitium and the capillaries is the basolateral side. I'll say that again. The side that faces the lumen is the apical side, and the side that faces the interstitium and the blood vessels is the basolateral side. So when you hear those terms, that's what they mean. So now let's talk about how reabsorption works and how we reabsorb different things. First, let's talk about how we reabsorb sodium, because sodium is a key player for reabsorption. Now, when we reabsorb something, first it has to cross the apical side, and then it has to cross the basolateral side. But the basolateral side drives the apical side, and you'll see what that means in just a moment. On the basolateral side, the side closest to the interstitium, the side closest to the blood vessels, primary active transport, the sodium-potassium pump that we learned about back in Bio 137. The sodium-potassium pump moves 3 sodiums out of the cell. It moves 2 potassiums into the cell. Now, remember, potassium is always leaking out of a cell. It leaks down its gradient. So movement of sodium is out of the cell. Now on the apical side, the side closest to the lumen, so right here, this layer, we were just talking about the basolateral side. Now we're talking about the apical side. On the apical side, The basolateral activity, like I just said, it creates the driving force. So it creates an electrochemical gradient for sodium. If we're pushing sodium out on this side, it's going to draw sodium in on this side. Now we'll see a bigger picture in just a moment, so don't worry that this is a really small picture here. So how does sodium get in on this side? Well, sodium channels, leaky sodium channels, and sodium co-transporters. Some sodium just leaks in, and in some cases, we have sodium co-transporters. Sodium co-transporters, think back to Bio 137. Sodium co-transporters are transporters, proteins, that let sodium come in attached to something else, like glucose, or certain vitamins, or amino acids. That means that not only are we reabsorbing sodium, but it allows us to reabsorb other nutrients. So let's take a look at that. Now, this is a big picture. There's a lot going on, so I'm going to spend quite a bit of time with it. Over here, this is that basolateral side. This is that side that is closest to the capillary, the interstitium. So here is one of those sodium-potassium pumps. It's using ATP, so it's pumping 3 sodium out and 2 potassium in. Now remember, potassium leaks out of the cell. It leaks out of the cell. Some of that potassium is just going to move into the bloodstream. We're going to reabsorb it. But most of that potassium just kind of goes through a circular route. It leaks out of the cell, it's pumped back in. It leaks out of the cell, it's pumped back in. And that just continues. But the sodium, as it's pumped out of the cell, it's carried away in the bloodstream. It continues to get pumped out of the cell, and it's carried away in the bloodstream. As it's pumped out of the cell and it's carried away in the bloodstream, what does that do inside the cell here? It creates a very low concentration of sodium. If there's a low concentration of sodium here, and there's a high concentration of sodium in the filtrate, what's going to happen? Sodium in the filtrate is going to want to leak into or diffuse into the cell. And it does. Some of it is going to come in through leaky tubules. Some of it is going to come in through co-transporters. And as that sodium comes in through a co-transporter, some of those co-transporters are bringing in sodium and glucose. Some bring in sodium and amino acids. Some bring in sodium and vitamin. As that sodium comes in, it's pumped out and it's carried away. The vitamins come in. They move through the other side and they're carried away. So here what we're doing are those things that were previously filtered, the sodium, the glucose, the amino acids, the vitamins. Our body wants to keep those. Here we are reclaiming them. We are reabsorbing them. Now the sodium, why is it so important that we are keeping that sodium? Well, what is sodium? It's the most important solute. And what do solutes do? Solutes suck. As we reabsorb this sodium, what do we think comes with it? Water. The water that's in the filtrate, we want to keep that. Remember we said we reabsorb about 99% of the water that we filter? Well, here we go. That water comes right through. This is called an aquaporin. Specifically, this is called an aquaporin type 1. Aquaporins are just little channels that let water through. The water comes through following that sodium and all of those other ions, all of those other solutes, comes right through the other side into the blood to be carried away. So that is reabsorption at the PCT. Now let's go back to tie that to this other picture here. That filtrate is moving through the tubule. Here, this is the wall. This is that cell we were just looking at. So everything was moving through in this direction. We could have just easily been moving through in this direction. But remember, this is one layer thick. This is that one cell we were just looking at. So the water that we were just looking at, it was passive. It did not require any ATP. The sodium required ATP, but all of this other stuff, it did not require ATP. And water is drawn to all of it. Water's drawn to sodium, it's drawn to any solute. And as those solutes are being reabsorbed, water just goes along for the ride. It passes through those aquaporins going towards wherever those solutes are. So passive water reabsorption. And water passes through aquaporins, those water tubes, those water channels. And there's two types of aquaporins. There's something called aquaporin type 1. Aquaporin type 1 are always there. They are found primarily in the PCT. They're found throughout, but primarily they are in the PCT, the proximal convoluted tubule. Aquaporin type 2 are what ADH produces, and they are found in the distal convoluted tubule and the collecting duct, primarily the collecting duct, but it is ADH that produces them. So there are very few aquaporin type 2s except when ADH tells them to be produced. Aquaporin type 1, always there, primarily in the PCT, well, and also the descending limb of the loop of Henle. Aquaporin type 2, only there, really, in the presence of ADH, in the DCT, and primarily the collecting duct. And remember, aquaporins, they let water through, but water goes wherever the solutes are. So these aquaporins, while they let water through, they're kind of useless unless there are solutes there to pull the water through those aquaporins. Now, another thing I want to talk about is something called transport maximum. Transport maximum is a limiting factor of proteins. Now, as things move through, let's just talk about the proximal convoluted tubule because that's where most things are reabsorbed. And all things have a transport maximum. I'm going to talk about glucose, but the same thing goes for vitamins and everything else, amino acids, but we're just going to talk about glucose. As glucose moves through the proximal convoluted tubule, it is reabsorbed. And once we get to the end of the proximal convoluted tubule, there are no more proteins that reabsorb glucose. There are no proteins that reabsorb glucose in the loop of Henle, in the distal convoluted tubule, or in the collecting duct. But that's okay, because as long as the person is healthy, by the time we get to the end of the proximal convoluted tubule, all of the glucose has been reabsorbed. Now, those proteins that reabsorb glucose, they work at a certain speed. And in the average healthy adult white male, that speed is enough that they can reabsorb all of that glucose. But let's say someone sits down and they eat an entire chocolate cake. Well, now they're going to have a lot more glucose in their body than they typically would have. All of a sudden, there's going to be a lot more glucose getting filtered, and they're going to have a lot more glucose in their filtrate. Those proteins that reabsorb glucose suddenly cannot reabsorb all of that glucose. They can't work fast enough. And by the time we get through the proximal convoluted tubule, there's still going to be some glucose left in that filtrate. What we say is we have exceeded transport maximum for glucose. That means that even though typically we reabsorb 100% of glucose, sometimes there's more glucose than those proteins are able to reabsorb. They can't work fast enough. There's not enough of them. The same thing can go for vitamins. Now, unless you are vitamin deficient, there's really no need to take extremely high doses of vitamin supplements. And as a matter of fact, it's common for people that work in hospitals to say something like, if you're giving your patient high dose multivitamins, really what you're doing is causing them to have expensive pee, because the vitamin, they're expensive to give your patient, and all that's happening is those vitamins are exceeding transport maximum and they're just peeing them out. So the other thing that happens is in diabetics, well, they regularly have high glucose in their blood, and their glucose is going to always exceed transport maximum. Well, glucose, remember, is a solute. So as glucose gets to the end of the PCT, well, now there are no more glucose transport proteins. Glucose is going to remain in the filtrate throughout the rest of the nephron. And since it is a solute, what's it going to do? It's going to suck. It's going to suck water towards itself. It's going to resist water being reabsorbed. That means that there's going to be more water in the urine. There's going to be more water in the filtrate. So the person is going to urinate a lot more. That process is called osmotic diuresis. And that is why people with diabetes mellitus urinate a lot. It's called osmotic diuresis. And that's because glucose exceeds transport maximum. So let's talk about what is reabsorbed. Now, right here we're looking at the blue boxes, and specifically right now we're looking at what's reabsorbed at the PCT. So this blue box right here, coming from the PCT, 100% of glucose and amino acids, 100% of most vitamins and nutrients are reabsorbed at the proximal convoluted tubule. 65% of sodium and water are reabsorbed at the proximal convoluted tubule. Most electrolytes are reabsorbed at the proximal convoluted tubule. So what's reabsorbed at the loop of Henle? At the descending limb of the loop of Henle, water is reabsorbed. At the ascending limb, of the loop of Henle, there is a thin limb and a thick limb. At the thin limb, there is passive sodium reabsorption, and at the thick limb, there is active sodium reabsorption. And actually, at the thick limb, it's active sodium, potassium, and chloride reabsorption. Now, the loop of Henle, we're going to devote an entire lecture to that in our next lecture. So don't worry about how this happens. We're going to devote an entire lecture to that next. Now at the DCT and the collecting duct. So let me preface this by saying what we've said so far about the proximal convoluted tubule, that happens all the time, regardless of anything. And the loop of Henle, it reabsorbs water and sodium, potassium, chloride always, regardless. But the distal convoluted tubule and the collecting duct, they change what they do based on the state of the body. So when we talk about what do they reabsorb, what do they secrete, it's going to be different from time to time. It's going to be different based on what does the body need. So by the time we get to the distal convoluted tubule, only about 10% of the original filtered sodium and chloride remain, and only about 25% of the original filtered water remains. 90% of that sodium and chloride have been reabsorbed by this point. 75% of the water has been reabsorbed by this point. Now, at the distal convoluted tubule and the collecting duct, which really, they are almost the same thing. They're not, but they're almost the same thing. Here, ADH causes water reabsorption. It causes urea reabsorption. And it does this in both the distal convoluted tubule and the collecting duct, but it has more effect in the collecting duct. And think back to when we were talking about the endocrine system. This is where diabetes insipidus has its effect. ADH is blocked in diabetes insipidus. So this is where that happens. Now, aldosterone, when aldosterone is being released, remember, it causes sodium reabsorption and potassium secretion. Why does it do both? Well, remember when we were talking about aldosterone back in endocrine, we said that it causes the insertion of sodium-potassium pumps. Sodium-potassium pumps reabsorb sodium, secrete potassium. And if we're reabsorbing sodium, chloride's going to follow it. So it doesn't directly cause reabsorption of chloride, but it does indirectly because chloride has a negative charge and we're reabsorbing positively charged sodium, negatively charged chloride follows it. Now, we said that ADH has more of an effect in the collecting duct. Aldosterone has more of an effect in the distal convoluted tubule, but it does have a little bit of an effect in the collecting duct. So, kind of a mini thinking cap question. Why do ADH and aldosterone always travel as a team? They're both you never have ADH release without aldosterone, and you never have aldosterone release without ADH. Why is that important? Well, if you only had ADH being released, we would be inserting aquaporins, but big deal, there wouldn't be any solutes to pull water through because aldosterone causes that. But if you only had aldosterone, we would be reabsorbing sodium, but there wouldn't be those aquaporins for water to come through. So big deal. You have to have aldosterone to have that pulling force, to have those solutes to suck the water through, and you have to have ADH to provide the pathway. ADH provides the pathway, the aquaporins. Aldosterone provides the pulling force, those sodium ions. All right, what else? Atrial natriuretic peptide, ANP. We talked about that back in endocrine. ANP, in some situations, inhibits sodium reabsorption. It does that by blocking aldosterone. So that inhibits water reabsorption, and we'll talk more about ANP in our next lecture. We'll talk about how it works, how it blocks water reabsorption. Parathyroid hormone. When we need more calcium, it increases calcium reabsorption. Remember back when we were talking about PTH? One of the things that it did was at the distal convoluted tubule, it increased calcium reabsorption. So what about secretion? Now we're looking at the green boxes. Tubular secretion. So secretion is just reabsorption in reverse. Something that was not previously filtered at Bowman's capsule. It remains in the blood, but we want to put it into the filtrate. So maybe there was just a whole bunch of it and it didn't get filtered. There was too much of it, so it stayed in the blood. Or maybe it was something that was bound to plasma proteins. Some things that were bound to plasma proteins, well, proteins are so big, they don't get filtered. So they didn't get filtered. Or maybe they had the wrong charge. Whatever it is, maybe it didn't get filtered. So we need to take it out of the blood and put it into the filtrate. So certain drugs, certain metabolites that are bound to plasma proteins, those get secreted in the proximal convoluted tubule. Urea and uric acid in the proximal convoluted tubule. We'll talk about that a little later in our next lecture also. Potassium. Potassium was previously reabsorbed in the PCT, but then it is secreted in the distal convoluted tubule and the collecting duct by aldosterone. So, pH maintenance. We'll talk a little bit about this now, but it's more important in our next chapter. So, mostly pH maintenance happens in the collecting duct. It's also a little bit in the distal convoluted tubule, but not very much. So, in the case of H+, when H+ is high, H+ secretion goes up. So when the body is acidic, that means that there is excess H+ in the bloodstream. When there is excess H+ in the bloodstream, H+ gets secreted. Also, increased chloride reabsorption will replace bicarbonate reabsorption when the body is alkaline. And when the body is acidic, bicarbonate is retained and generated. So when the body is acidic, we can generate bicarbonate. And remember, bicarbonate is a buffer, so it can help fight that acidity. Now, We will talk a lot more about this. It'll be more important when we get to our next chapter, because that's really what the topic of the next chapter is how does the body maintain pH. So right here, don't worry too much about it. We're going to talk about how this ties in our next chapter. So we'll come back to this. But for right now, that's going to be where we take our break. We will, in our next lecture, talk about the loop of Henle and something called the countercurrent mechanism. It will be a bigger lecture with a lot of drawing, so have some paper handy for the next lecture. All right, I will talk to you in our next lecture, and until next time, take care.